Studying graves can allow us to gain intriguing insights into many aspects of historical lifestyles. But how do we gain information from a grave? Previously, the main focus of studying graves surrounded looking at the macroscopic features of the grave. However, Matthew Pickering et al. Demonstrated that molecular and microscopic contents of the grave can also be useful in these studies. They studied two sets of skeletal remains, with one set belonging to the Roman era and the other set belonging to the Anglo-Scandinavian era. As these skeletons decomposed, the organic composition and microstructure of the surrounding soil had been altered, giving distinct signatures that can be preserved over long periods of time. Pickering et al. Investigated these signatures using analytical chemistry and by studying the structure of the soil. The techniques that were used were microscopy to analyse the structure of the soil, chromatography to separate the organic components based on their polarity. The more polar fractions were then analysed using gas chromatography and mass spectrometry, and liquid chromatography was also used. The soil was found to contain a variety of organic compounds. These included triacylglycerols and their hydrolysis products, diacylglycerols and monoacylglycerols, which were present due to the breakdown of adipose or fatty tissue. Fatty acids and aldehydes, which varied differently based on anatomical location, showing that bone could restrict water movement in some areas like the skull, creating areas of low oxygen concentration that favour the reduction of the fatty acids. Hopanols, which indicate the presence of bacterial populations, which are present due to the decomposing body providing a good source of organic substrate, and cholesterols and some of its derivatives. Cholesterol and its derivatives proved to be a valuable source of information in the graves. Both graves showed higher concentrations of cholesterol in areas associated with more decomposition, suggesting a link between cholesterol and bone degradation. Furthermore, considering the ratios of different isomers of reduction products of cholesterol allowed the fecal contamination of the Roman grave due to cesspit leakage to be accounted for. The major isomer is present due to production by plants and soil, and the minor isomer is present in the gut of many higher animals, and is therefore the major isomer in human fecal matter. The microscopy analysis revealed an abundance of spherulites, which are small rounded bodies, within the Roman grave. The spherulites were able to show changes in redox conditions based on changes in their microscopic structure. Analysis of the soil showed that many different compounds can be used to learn about a grave, even once decomposition has started to occur. For example, the link between cholesterol and bone degradation could be used to identify burial locations, even when the remains have been poorly preserved. This paper showed the promise that microscopic and molecular analysis of graves holds, and further research into this field could uncover a whole new skill set for archaeologists and chemists to utilise when studying burial sites.